Thanks so much, guys, for coming. Today we are doing male reproduction. So the first of the lectures for the final exam. So we only have 4 packets for this one, and it's all to do with the birth, with reproduction, because we will talk about male reproduction in this lecture, and then female reproduction, and then we'll talk about pregnancy, and then birth control into the end. So we have, not so much material to cover though. So in this first part, we'll talk about the male reproductive anatomy, and then we'll talk about the physiology of it and how it is controlled via hormones, some of which we've already seen when we talked about the endocrine system, right? And then we'll just finish off talking about the effects of testosterone. All right? So, functions of the reproductive system. This is for both male and female, and then we'll go into just the male, and then the next we'll do the female. All right. So to produce offspring, when the male gamete and the female gamete come together, okay, then you form a new individual, and this grows into the female reproductive tract. And so, on top of that is production of sex hormones, estrogen for the ladies, for the females, and then testosterone for the males. All right. So the primary sex organs will be the organs that produce the gametes for each sex. So it will be testes for the male, it will be ovaries for the female. Everything else is all accessory organs. Okay? Everything else is all accessory organs. Okay? And therefore, the primary sex organs produce the hormone for that particular sex. And actually, androgens are, male hormones, of which the most potent is testosterone. Okay, it turns out that estrogens, they are precursors androgen. Okay, and even in males, you find just a little bit of estrogen, which is produced by the cis testicular cells, just a little bit. But the dominant one, of course, will be the testosterone. Okay, all right. So females do the estrogens and progesterone, okay, which features a lot during pregnancy. All right. So of course they develop the reproductive organs for both sexes. These androgens for males, of which the major is testosterone, and the estrogen and progesterone for females. And of course, sex drive and sexual behavior is governed by or controlled by those androgens. So that's why when you have low T for males, they lose sex drive, they lose, they're generally lethargic too, because and their mood is down as well. Those hormones, they elevate your mood as well. And then of course growth and development in other organs. And we'll talk about the effects of testosterone, and you see that it's a cause of vascular development in males, right? So we'll talk about that. And then, of course, everything else other than the ova for the female and the testes for the male is accessory. And they're called genitalia, all right, external genitalia, and ducts and glands, that produce secretions and so forth, especially for the male. All right, so now we move particularly to just the female. The male reproductive organ. Of course, the testes will be, they are located or housed outside of the core body, so that the testes are about 3 or so degrees lower than body temperature. That's because this is the optimum temperature for sperm production. Okay, so that's why they are in a sac. But actually, they develop very close to the kidney as a retroperitoneal organ behind the peritoneum, and then it comes through, and some of the peritoneum becomes some of the coverings of the testes. Okay, we'll talk about that in a little bit when we talk about the descent of the testes. All right. So sperm are delivered, through a ductwork of, a system that conducts to the outside, ending with the urethra. So it's a duct system that comes from the testes, starting with the epididymis, and then it goes up, and we'll talk about all those. Then the vas deferens, which is about 45 centimeters long on both sides, and then it goes into the ejaculatory duct, where it mixes with secretions from the seminal vesicles located behind the bladder, right? Kind of sort of dilates the vas deferens, and then it goes into the urethra, which has three sections to talk about. The prostatic urethra, which goes through the prostate, then the membranous urethra that goes through the floor of the pelvic cavity, and then of course the spongy urethra, so-called because one of the erectile tissues through which the urethra goes is called the corpus spongiosum. All right? So those are the ductwork, and then another group of these accessory organs will be the glands, really, that secrete all the secretions. We'll talk about each one of them, starting with the prostate, the seminal vesicles, and the bulbourethral glands, right? And we'll talk about all the secretions from each of those, all right? And then, of course, what are the functions for the male particularly is to produce the sperm cells, okay? And then, of course, to then sustain their, viability and transfer them to the female, all right? And then, of course, to produce the sex hormones like testosterone, all right? So, gotta know, of course, the functions of each of the sexes, the reproductive systems of each sexes, and then, of course, the components. For the male, of course, this is all that is, starting with the testes, epididymis, vas deferens, and then those glands, seminal vesicle, prostate, and bulbourethra, and then the urethra, and then the female, the ovaries, the oviduct, and so forth. And we'll talk about that later. The uterus and the vagina, not the urethra in this case. All right, it's different. So we'll talk about that, and then of course the mammary gland. All right. All right. So like I said, we'll start talking about the anatomy of it, and then we'll talk about how it works. All right. So the testis, primary organ. Okay. And then of course the duct. Okay. The epididymis is where epididymitis is plural. Okay. The sperms mature, and we'll talk about that in a little bit. They're producing the somniferous tubules, and then they mature for about 16 or so days in the epididymis, and then the epididymis is connected to the vas deferens, or ductus deferentiae. Ductus deferens. Same thing, okay? Vas deferens, ductus deferens is the same thing. Okay? Vasa differentia is same as ductus differentia. This is plural, by the way, because there's two of them, one on the left, one on the right. And then urethra, of course, the one that's split into three sections, we'll talk about it. All right? And then accessory glands, seminal vesicle, located posterior to the bladder and sort of on the inferior part of the bladder. And then the ducts feed into the ejaculatory duct. And then the prostate gland. The prostate gland is kind of sort of a walnut-shaped, donut-shaped organ that literally surrounds the urethra inferior to the bladder, okay? Which is what is called. And then these are peanut-sized glands on either side, just below the prostate, that feed into the membranous urethra and produce an alkaline fluid. When the male is first sexually, stimulated, this is the secretions that come out first, and you see them, the colorless kind of secretion. That's a slimy, mucus-like secretion for lubrication, of course. But one of the primary effects of that is to neutralize acidic urethra, right? Because urine is acidic, and most of the time the urethra is conducting urine. So, and it makes sense that it's the first secretions you see. Whenever a male is sexually aroused, that's what you secrete first to neutralize the urethra before you get the sperm coming out. Otherwise, they're going to be killed a little bit. Some of it at least So you need the secretions first. And of course, some supporting structures like the scrotum and the penis. The penis is there just to become erect so that you can insert it into the vagina to deposit the sperm. So it's a supporting structure. And the scrotum, of course, we said it's down there because the optimum temperature for sperm production is 3 degrees less than core body temperature, right? And then, of course, all collectively known as male genitalia, all right? And then, of course, there is the testes, there is the vas deferens. I mean, the vas deferens is here. This is the epididymis split into the head, the body, and the tail. It's got those 3 things going on it. And then, of course, you got the seminal vesicle behind the bladder, and then this is the prostate right here. Okay, the prostate. So therefore, this is the prostatic urethra. All right, and then this part here that joins to the spongy corpus spongiosum is the membranous urethra, and then the spongy urethra finishes it off. Okay. And then the penis is made of 3 columns: the corpus cavernosa, which is 2, one on either side, and then the spongiosum on the bottom. Okay, so if you kind of take a cross section of the male penis, this is kind of what you find. And when it's erect, it's very evident. You can see, well, it doesn't appear like a clover like that, but you can see the pillars, but this is an exaggerated diagram just to show you the 2 corpora cavernosa and then the spongiosum with the urethra in it. All right. So there it is, right there. You get the corpus cavernosa, 2 of them, 1 corpus spongiosum, which is singular, right? And those are the erectile tissues of the penis, all right? Epididymis, and then because this is one of those questions that will lead itself to those kind of tracking and matching kind of questions for this coming exam. When you line up the ductwork or how sperm travel from the seminiferous tubules all the way to the outside, So those are the things I'll be needing you to track when we do this. So the scrotum, of course, it's a pouch of skin and connective tissue that suspends the testes below, the pelvic cavity, right? And it has 2 muscles. Okay, the dartos muscle is wrinkles up the skin of the scrotum to elevate the testes whenever they're in a cold environment. If it's way too cold, the testes are raised up so that they are close to the core body temperature. All right, and then another muscle that is literally on the spermatic cord is the cremaster muscle. You see, the spermatic cord comes down like that into the scrotal sac, and then it surrounds the testes. Okay, so the cremaster muscle runs down here. There's connective tissue on the outside, fascia on the outside, and then the cremaster muscle runs like that. So when it contracts again, it raises the testes up and down, depending on the temperature. If you see a guy, in a hot day, they are really saggy, and the dartos muscle, you know what I'm saying? That's what happens, I'm telling you. Or if you go into a hot bath, it really comes down so that it runs away from the core body temperature, right? So, and then when it's cold, they get wrinkled up and they're all close to the core body temperature. So, gotta know these muscles and gotta know their location, all right? And what their function is, of course, is to ensure that testes are about 3 degrees lower than core body temperature. And that the reason is because sperm production is optimum at about 34 degrees C.
So if you're trying to get pregnant with a couple, you want your man to wear boxers or something.
Yeah, that lets, and also don't be like sitting in a hot tub and in a jacuzzi, you know, and be like toasting them like that.
Yeah,
then
romantic.
sperm production goes down, but it's not really good for reproduction. Okay, so you might want to take showers, more showers, warm showers, not hot showers. Don't be soaking yourself all in the shower and steaming up, okay? Because then it's not good for sperm production. You know, that's what it is, right? Take a cold shower. All right, so gotta know the scrotum, of course, what it is, and then of course the tissues that surround it, you know, and the muscles, you know, and what they're for. All right, so so the testes, we kind of sort of making a cross section of the testes, and we're gonna talk about the duct work and all these things. But before we do that, we're gonna talk about how the testes start off by the kidneys and come through the peritoneum, and then suspended in the scrotum. All right. So the tunica vaginalis. Remember, I said it's a retroperitoneal organ. So this will be the peritoneum covering the, you know, the serous membrane demarcating the abdominal pelvic cavity, and then you've got the kidneys here developing, and then you also got the little testes developing here. So when they go through here, this peritoneum, it goes through, and then it will surround the testes, because the testes are going to go through the inguinal canal, and that becomes the, vaginalis, the tunica vaginalis, the peritoneum does. All right, and then the fibrous capsule on the outside then forms the tunica albuginea, which is kind of whitish, because albuginea from albino, you know, white, so it's white, very tough connective tissue that surrounds it on top of the tunica vaginalis, right? And the tunica vaginalis comes from the peritoneum, okay? So the tunica albuginea, what it does, it divides the testes into columns, into sort of cone-shaped columns like this. You know what I'm saying? It will be on the outside, and it will invaginate inwards and divides it into lobules like this. All right. And the sections of the tunica albuginea, that go in are called septa. You know, so that in there you'll find the seminiferous tubules in there. Okay, 1 or 3 seminiferous tubules inside of that. All right. So if you take the seminiferous tubules from the left and the right testes, and you string them into a length, it's about half a mile of seminiferous tubules. That's long, right? That's where you produce a whole bunch of sperm in there. Okay? So the septa then divide it into about 300 to 400 lobules, right? With the 1 or 2 seminiferous tubules in there. So, so the septa come from the tunica albuginea on the outside. Right. Just like some of the other organs, like the thymus, they are divided into the lobules by the connective tissue. And these ones are called, you know, the septa. Those are columns of connective tissue. So moving on, so we talked about how you have about 300 to 400 lobules in it, 1 or 3, 4 seminiferous tubules. Okay, and seminiferous tubules are kind of sort of, they're little ducts, right? Like this, and they have cells like that called Sertoli cells. Okay, that surround. So the Sertoli cell will span the basement membrane on the outside here. Okay, and then you have another Sertoli cell. They are big cells, and then the lumen of it. That's where. So this is like the seminiferous tubule right there, and then between these will be spermatogonium and so forth in different stages of development. And then by the time it gets here, it's a sperm, right? And then the sperm will be inside the lumen, okay? So the seminiferous tubule has got little ducts that, then called tubulus rectus, that the sperm go to after they are produced, and they are immature though, all right? And then there's like a part that looks like a net called the reti testis, like net reti testis. So it kind of looks almost like this. And I'll show you a nice picture, like a net, something like that. And then it's got little, and then so you have the tubulus rectus and then the seminiferous tubules in the lobules. Okay, so sperm starts in the seminiferous tubules and then tubulus rectus and then go to the reti testis. All right. Okay. And then there are sort of little straight tubules, and I'll show you a picture of it, called efferent ductules. Okay, that will lead into the epididymis. All right. So you get the tubulus rectus from the seminiferous tubules, and then you get the reti testis, which is how the sperm start from the seminiferous tubules, tubulus rectus, reti testis, and then efferent ductules that go to the epididymis. And then the efferent ductules will lead the sperm away from the testis, right? Okay, so if I was to, what's that?
E for exit.
E for exit, yeah, efferent ductules.
That's a good way to remember it.
Yeah, true, E for exit, yeah. Because sometimes I say, can you track this production of a sperm? Sometimes I even go as far as until it enters the female reproductive tract, ending where fertilization occurs in the oviduct. Sometimes I do that. That would be a nice kind of sort of 10-point question where you're tracking the structures the sperm is traveling through. Okay? So starting.
Your question might be
sometimes
all the way from one
sex to the other. Sometimes if I do half, maybe I might just do sperm until they come out of the urethra. You know what I'm saying? So it will start off in the seminiferous tubules, it's produced, then it goes into tubulus rectus, tubulus rectus, and then the reti testis, the efferent tubules, and then of course the epididymis, vas deferens, ejaculatory duct, and then urethra, right, and then outside. Okay, so just remember how the sperms, the four different tubules, they go through while in the testis, okay, until it ends up in the epididymis via the efferent ductus.
So it goes 1, 2, 3, 4.
Yeah, it goes seminiferous tubules, then it goes tubulus rectus, then it goes reti testis, then efferent ductules will go to the epididymis. So it only goes through 4, starting with the seminiferous tubules, and then surrounding the seminiferous tubules, okay, will be the interstitial cells. And then you have another seminiferous tubule here, okay, and then you have more of these. Those interstitial cells, they produce testosterone. All right. So sperm production in the seminiferous tubules, and then testosterone is produced by the interstitial cells, sometimes called Leydig cells. Okay? Leydig cells. Okay? Those are the cells that produce testosterone. And later on we'll talk about luteinizing hormone, which stimulates the interstitial cells to produce testosterone. Okay. Luteinizing hormone is also called interstitial cell stimulating, right? Because of the interstitial cells that produce testosterone.
I think in the text it said that they found that there was no difference in them, so they just went to LH for both.
Yeah, because LH kind of causes ovulation actually. Just before a female ovulates, there's an LH surge, luteinizing hormone, a spike in LH. So those females who are having trouble getting pregnant, that would be one of the things you track is of course, besides the temperature change, but the LH spike, you need a physician to help you with that. The temperature change you can get quickly, but just before ovulation, boom, LH spike. All right? So just know these structures and which cells are doing what, which structures are doing what. Here, the seminiferous tubules, production of testes, and the seminiferous tubules have these Sertoli cells that I'm going to talk about, or sometimes called Nest cells as well. So these are the seminiferous tubules, like I was saying. You've got this Sertoli cells, sustentacular cells, nest cells, and so on. They got many names. Okay, these sustentacular cells are those cells that I was drawing that span the thickness of the seminiferous tubule. And then the lumen, that's where you have the sperms inside of it. All right. So you start off with the spermatids. The spermatogonia, then primary spermatocytes, secondary spermatocytes, spermatids, and then spermatids are the ones that differentiate into sperm cells. Yes, spermatozoa. Okay. Because the sperm cell is not circular like the cells that start it off. The spermatogonia and all these other cells are circular, but the sperm is got a separate three sections actually: the head, the midpiece, and the tail, the flagellum, really, which forms the helps the motility wise. So this is what we were talking about earlier on. We're talking about how you have the seminiferous tubules, okay, divided by the septa here, the connective tissue, and then seminiferous tubules, the tubulus rectus here, and then you see this retitestis, which looks like a net. The retitestis collects it, okay. So seminiferous tubules and tubulus rectus, and then the retitestis, and then the efferent diffuse, okay. And then 4. So it goes, sperm goes through those structures before it gets out, into the epididymis. Epididymis, which is the head, body, and tail. See that? It's really kind of evident: body, head, and tail. Okay? And then that's the ductus deferens. All right? And then, so here you've got the acrosome, which contains enzymes, one of which is higher hydroxylase. They're proteolytic enzymes and so forth that will break through the wall of the ovum, the egg cell, ovum. All right? All right, so that's that. And you can see the seminiferous tubules with the sustentacular cells that support and sort of nurse the developing sperm cells. Okay? So reproductive system. So like I said, testes starts off as a retroperitoneal organ, very close to the kidneys, and then they descend through the inguinal canal and then pick up the peritoneum, which becomes the tunica vaginalis, all right, and then it goes down into the scrotal sac. All right. There's a structure called the gubernaculum that kind of guides them to go down, in the correct path, right, to get into the scrotal sac. All right. So the tunica vaginalis, which is the peritoneum, right? The peritoneum is got a visceral peritoneum and the parietal one, and the parietal one is becoming the tunica vaginalis because it becomes internalized, and the outside one becomes the tunica albuginea, right? And the good thing is that as the testes are traveling down, it is protected from friction, so that it's not brushing against the other organs as it's going through in the inguinal canal. So it serves those processes, those functions too, right? So that's the tunica vaginalis. So you got it as a retroperitoneal organ, and it goes through, okay, and then it will travel down the. See, the gubernaculum is the structure that kind of guides the descent of the testes, so that the testes go into the correct path and end up into the scrotal sac. All right? And then all this peritoneum that it goes through, okay, kind of comes down here, and then this, you see, process vaginalis, which is an extension of the, that just follows the gubernaculum as the testes are descending, becomes the, the outside layer covering the testes here, all right? And this part here kind of sort of degenerates, all right? So that you have the abdominal pelvic cavity intact here, it kind of cuts off, and then this, and a little bit of the gubernaculum is left here. It just becomes a ligament, okay, that was guiding the testes down, right? then of course the ductus deferens forms within the inguinal canal. The ductus deferens carries the sperm cell to the ejaculatory duct. All right. So you just know that of course the sperm, the testis starts as a retroperitoneal organ behind the peritoneum near the kidneys, and then descends through the inguinal canal, guided by the gubernaculum, which is the kind of stalk structure that goes down into the scrotal sac. All right, and the peritoneum becomes the tunica vaginalis. All right, and that's it. Really. That's all I want you to know about the descent of the testes. There's a condition called cryptorchidism, which the testes do not descend into the scrotal sac. Okay, and those females, those males are impotent. They cannot produce sperm cells. Why? Because the testes are being toasted up there in body temperature. Okay, you could have one or two of them descending, or both of them not descending. So then that will be the sum. Okay. If one of them descends, you might be able to produce enough sperm count, but you might also be impotent because the sperm count may be low. Right? Spermatogenesis now, the production of sperm cells. All right, pretty much on the inside the seminiferous tubules, which is made up of those Sertoli cells, or the nest cells, or the sus tentacular cells. Right? We start off with spermatogonium, then it just divides up into two, right? One of them becomes a primary spermatocyte, and then the other one continues as a spermatogonium, right? Makes sense? Because if both of them become a primary spermatocyte, then there will be no cell line left to continue to generate more. Okay, so that's what spermatogenesis is, simply. And then the primary spermatocyte will become the secondary spermatocyte, okay, when it divides the first meiotic division, then it's now haploid, all right? And then when it does the second meiotic division, which is simply mitosis, increases the number, then it becomes a spermatid. And then you're done. At that stage, all you need is to differentiate the spermatid, which is the last step in spermatogenesis, to form a sperm cell. And you're done. Good. So sometimes in the exam I ask you to define spermatogenesis. I mean, not to define spermatogenesis. Sometimes I ask you to define it, but to talk about the processes that lead to the production of sperm cells. Okay, so just start with this spermatogonium, right? Divides into two. One becomes primary spermatocyte, which continues to develop, and the other one just remains a spermatogonium and continues to divide. So it produces a type A and a type B cell. Okay, the type B cell is the primary spermatocyte, which then goes through the first meiotic division, which is a reductive division, halving the genetic material to a haploid cell. Then you have a secondary spermatocyte. Secondary spermatocyte then goes through the second meiotic division, which is simply mitosis, just increases the number, okay, so that you end up with 4 sperm cells, right, from that one. And then spermatid is the last step before it differentiates into the, okay. So that's what I really want you to know about this, and I'll list the steps for you in like a diagram so you can kind of visualize it. But that's pretty much this stage, right? The last stage where a spermatid differentiates into an actual sperm cell, that's spermiogenesis. Okay? Spermatogenesis is the whole thing, the whole process. That's what I'm saying. So don't confuse this guy. Spermiogenesis is simply the differentiation of a spermatid, which is a circular cell, to form a sperm cell with those 3 regions: the head, the tail, and the meat piece. Right? Okay. Spermatogenesis is the whole process, starting with the spermatogonium. Okay. All right. So spermatogenesis, the spermatogonium on the outermost, close to the these are developing between the sus tentacular cells, and the sus tentacular cells do the nursing and so forth. Okay, and then this divides into 2, and then this is the primary spermatocyte. All right, and then goes through first meiotic division. All right, then secondary spermatocyte. Okay, goes through the meiosis 1. Meiosis 2 forms the spermatid, is this one, all right? And then this one, then spermiogenesis, this is spermiogenesis, okay? Which is differentiation from a spermatid, a secular cell, to the, in its haploid, to the sperm cell. Okay. So this is the type A and type B cell. The type A cell continues as a spermatogonium and continues to, finish the cell line, so you produce more spermatogonium, and then it divides again into two. One becomes a sperm, primary spermatocyte. That's the type B one, and the other one becomes the, So the first one is just a mitotic division to increase the number of cells, right? So it's just mitosis, and the second division is meiosis, which occurs in two stages. Meiosis 1 and meiosis 2. So this is mitosis, which is a multiplication division, right? It just multiplies into, this is diploid, 46 chromosomes, all right? And this is, 46 chromosomes, and then after the meiosis 1, this is now 23 chromosomes. 23 chromosomes. Right? So just remember that little diagram there instead of what's written here, because it will be harder to remember that, right? See what I'm saying? So that is all that, all right? And then, of course, the last stage is the spermiogenesis, which is the differentiation of a sperm cell, a spermatid into a sperm cell, all right? So again, to beat a dead horse, you know what I'm saying? That's the same things, okay? Primary spermatocyte from the spermatogonium, secondary after haploid division, and then the meiosis 2, and then you form a spermatid, and then the spermatid differentiates into a sperm cell. And that's it. Okay? So this is repetitive. Again, same process, guys. Now only picture form. Okay? You've got your spermatogonium, 46 chromosomes, divides the first meiotic division, produces a type A, type B daughter cell. The type B daughter cell is the primary spermatocyte, which then kind of sort of becomes a primary spermatocyte. It's kind of like a one-stage thing. You know what I'm saying? The primary spermatocyte is one that divides into the secondary spermatocyte after the first meiotic division, which is a reductive division, forming a haploid cell. Okay, a haploid cell here. You see that we form two of them. Okay, and then each one will divide again mitotically this time, but via meiosis 2, which is simply a multiplication division. And then you're just multiplying what you had in the first place, which is a haploid cell. And then you form a spermatid. Okay. Then the spermatid will then go through spermiogenesis and differentiate into a sperm cell. All right. And then the sustentacular cells are between all these developing cells. And you see, this is a huge sustentacular cell. See, this is a big nucleus. And then you see these tight junctions between the developing sperm cells. They form the blood-testis barrier. Okay? They form the blood-testis barrier because sperm cells are not present when the immune system of the individual is developing, okay? Because before puberty, before you're producing enough testosterone, you do not have any sperm cells. So your immune system learns the MH complexes of the existing cells. You see what I'm saying? So the spermatids, there will be brand new cells that are actually foreign to the body. So if they ever get into blood, they can elicit an immune response, and you start killing your own sperm.
Like women in Rh negative blood type.
Mm-hmm. Same thing. Yeah, that's the same thing. Because the fetus is a foreign body. That's what it is. That's why the female immune system is depressed during pregnancy, to try and avoid, expelling and literally killing off the developing offspring. So same thing, because these cells were not there, okay, when the individual developed in the uterus and all that, and they only started to be there at puberty. So they are actually foreign cells. The body cannot recognize them as their own, because when it was learning all these, the cells of the body, they were not present. So it thinks that, whoa, that all the cells we need, anything else that comes is foreign. So that you need the blood-testis barrier. That's for me. And I will talk a little bit more about it when so these are the processes of spermatogenesis, right? So that's 5 points in your question.
What's the factoid question?
The spermatogenesis, starting with the spermatogonium and ending with the sperm cell. And the last step is just talking about how the testicular cells are, providing all the support and nutrients for these developing sperm cells, right? Okay, so spermatogenesis, of course, forms the sperm cell, which I said is step 3. The head, which contains the genetic material, the 23 chromosomes, also contains the aplosome, which contains hyaluronidase and those hydrolytic enzymes that will break through the egg cell. And then the midpiece has got a whole bunch of mitochondria for what?
Energy.
Energy to swim through the female reproductive tract. All right, and then of course the tail helps with swimming, to swim out. Only the head enters. Okay, the head and the midpiece enters, and the tail just falls off. It doesn't enter the egg, because all you need is the head piece with the genetic material.
Once you're in, you're in.
Yeah, that's it. The tail doesn't. The tail is just there for motility. It doesn't have anything else in it. Okay, so it's not needed. So it doesn't even go into the egg cell. Only the head does. Okay, so just know, the basic structure of a, sperm cell and what's located in those bits, which is very obvious and more intuitive than anything else actually. And then spermatogenesis, of course, the sus testicular cells, we were talking about it. Like the last step in spermatogenesis talks about how the sus testicular cells will nourish these developing sperm cells on top of producing tight junctions, which will shield these developing sperm cells from the immune system. So it forms the blood-testis barrier. Two very big, important functions, okay? All right? So nourishes the developing sperm cells, forms tight junctions that form the blood-testis barrier so that they don't go, any antigens of the sperm cells go into the blood, then you start to launch an immune attack against the sperm cells, because they are foreign. So this is simply talking about how these sperm cells did not exist until puberty, because at puberty, that's when you release a bunch of testosterone, which then stimulates the sus testicular cells, right? And then produce, these sus testicular cells, sometimes called Ness cells, sometimes called Sertoli cells, from the Italian scientists who discovered about, who shed more light on this. So the hormones these testicular cells produce, okay, are androgens, okay, androgens, and guess what? Estrogens. Just a little bit, okay? Others become human. And then a hormone called inhibin. Inhibin. Just by the name of it, what does it do? Does it stimulate or inhibit? It will inhibit production of testosterone, okay? Because testosterone, will cause these cells to produce sperm cells. So when you've produced enough in a negative feedback fashion, it goes back to the hypothalamus and the pituitary, so that you don't produce the gonadotropins, LH and FSH. FSH causes the production of more sperm cells. And I'll show you how the hormonal control is in it, okay? So those are the hormones that the sus testicular cells produce. Inhibin is a negative feedback on testosterone. T is for testosterone. Negative feedback to produce less testosterone so that you don't produce sperm cells. All right? Spermatogenesis, I almost feel like jumping this, but because it's the same processes of how the spermatogonium becomes. Do you want me to play it, or do you want to play it later on? You want me to play?
I'll play it. Why not?
Yeah, why not? Listen to it. Listen to it.
Spermatogonia are the cells from which sperm cells arise. The spermatogonia divide by mitosis. One daughter cell remains a spermatogonium, and the other becomes a primary spermatocyte. The primary spermatocyte divides by meiosis to form secondary spermatocytes. Secondary spermatocytes divide again to form spermatids. The spermatids differentiate into sperm cells.
Isn't that cool? I like that. So that's spermatogenesis in a nutshell. You could
I
listen
like
to.
how simple that is.
Yeah, you see what I'm saying? It just starts, spermatogonium divides into daughter cell, B and A.
It's not always that clean.
What's that?
It's not always that clean.
It's not always that clean. Yeah.
Sometimes you get double heads and double tails. And
Yeah. That's where you get your body, because it's producing this at a very, just amazing rate. So your body will edit and kill off the bad ones. So if your body has not got this screening processes wrong, then you end up with a whole bunch of sperm that are ineffective, and that lowers your sperm count and you become impotent. Because you need a lot of sperm cells to make it to be fertile. That's why you produce millions of them. Like in ejaculate, you have like 3 or so million, but 4 millions. Okay, so now we're going to talk about all these ducts. Okay, like the epididymis. I want you to know the function of it. So it's immediately on top of the testes, right? Those efferent ductules are leading to the epididymis, epididymitis for plural. And this is where the sperm cells mature. Okay, and I just want you to know, of course, the structure of its basic structure. It's got the head, the hand, the body, and the tail, right? And the sperm cells take about 2 weeks at least, 12 to 16 days to mature. When they are produced in the sustentacular, not in the sustentacular cells, but in the seminiferous tubules, they are non-motor, they are not active, the tails don't work, nothing works. It's just a structure, it's a shell, okay, with the goods, the 23 chromosomes. Then it goes in here. That's where all the action happens. They become active. They become the motility, and they become competent so that they can actually go through the egg. All those things are happening. You see, the acrosome matures. Without the acrosome, you got no enzymes to go through the walls of the egg cell. So you need that. Okay, so this is where it all happens. Okay, back in the sustentacular cells, in the seminiferous tubules, you're just making a shell, and then the ability to fertilize the ovum develops in here.
So
Those
it's kind of
2
like
weeks.
programming.
Yeah, it's programming it, it's activating it, and so forth. It's activating it and programming it, and giving it the ability to swim.
It's like we had the car, now we're adding the battery.
Yeah, it's like that's where you learn to swim. If that's your sport, you first learn and then you go to the Olympics. When you have sex, then that's the Olympics, it goes up. Otherwise, it won't be able to swim, it won't be able to fertilize the egg, it won't be able to do anything if it doesn't spend those 2 weeks in the epididymis. Okay, so I want you to know just the functions of the epididymis, what it's doing, how long it's taking, okay, and what abilities are being given to these sperm cells. The ability to swim, the ability to fertilize an egg, to acrosome, it becomes, and they're being activated, okay? The flagella now becomes able to move, okay? So know those things and know what the purpose of the epididymis is. It's just the basic structure, okay? So the epididymis leads to the vastus, okay? The ductus deferens, okay? So we get the epididymis with our head, body, and tail, and then, okay, boom, 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 there you go, okay? So look at these diagrams later on when you're, going through these things, so you can actually see the structure of these things. Kind of helps you to understand their function too. You know what I'm saying? So the ductus deferens, what it does, it simply conducts sperm cells. So the ductus deferens has got circular and longitudinal muscles, okay, layers of them. All right, and the ductus deferens, what it does, is that it's about 45 centimeters long, okay? So on either side, it goes like 45 centimeters, kind of long. So it kind of goes around like this and comes back. So just behind the bladder, where the seminiferous vesicles are, it will dilate, it will become bigger and thicker. And that's the ampulla, okay? And then the ampulla will join the ducts of the seminiferous vesicles, and then that becomes the ejaculatory duct. Okay. So that's what there is to it. There's a procedure called vasectomy in the males where they ligate, they cut off, and switch off. Yeah, it's a snip, snip. That's why when we're in the lab and I'm saying snip, snip, I'm thinking of vasectomy, which I'll never do. That doesn't make sense. With all the an amount of contraceptives there, oh my God. But anyway, this is a very effective because it's almost 100%. Because pretty much you're cutting the source, you're not going to get any sperm cells coming through. Okay, but you got the 45 centimeters of vas deferens where you had previous sperm in there. So that's why after you have it, you take about 2 weeks before you can actually have sex.
6 to 8 weeks.
Yeah, because 45 centimeters, that's almost 90 centimeters. That's a long meter. That's like a meter of sperm cells, all in that vas deferens. So you've got to, ejaculate a few times, more than a few times, to get rid of all the sperm cells before you are no longer, fertile.
You don't really feel like having it right afterward either.
What's that?
I said you don't really feel like having sex right after.
Yeah, because there's pain, which is one of the only side effect of it is the pain. That's why I'll never do that. You see? But anyway, it's effective.
How many kids do you have?
Okay. Okay. So that's
How many children do you have?
I got 3, so I got enough already. Okay, so I did my thing. We got the vas deferens going up, okay, 2 centimeters, and it goes to the back of the prostate where the seminiferous vesicles are located, all right, and then it dilates into the, you see what I was saying? It's got all these muscles. It's almost like the the GIT, like the small intestine, okay, because it is that peristaltic movement which then conducts the sperm up, okay, so the testicular secretions which are containing the sperm. So you've got the you see, all this is muscle. Okay? The longitudinal on the inside, okay, and then the middle circular, and then another longitudinal on the outside. All those contract to form that peristaltic movement that will guide the sperm up to the ejaculatory duct. Okay? So we talk about the ejaculatory duct. You see, the vas deferens, dilates into the ejaculatory duct. Okay, so the spermatic cord that I was talking to you about is got the ductus deferens. It's got the lymphatic and blood vessels all in there, and then it's got the cremaster muscles and the connective tissue surrounding it, the fascia on the outside. All that becomes the spermatic cord. Okay. So the spermatic cord, you get the blood vessels, arteries, and veins going down and up the ductus deferens, okay? And then surrounding it will be the cremaster muscle, the one we talked about, that raises the testes up and down, and then the connective tissue on the outside, which is fascia, okay, external and internal fascia that flanks the cremaster muscle, and that's all that is the spermatic cord. Okay, so the external spermatic fascia, then you got the cremaster muscle that raises the testes up and down, and then the internal fascia. So the cremaster muscle is flanked between external cremaster, internal fascia. Then inside of that, that's the covering of the spermatic cord. Then you get the blood vessels, the lymphatic vessels, and then the ductus deferens. Okay? And then the spermatic cord passes through the inguinal canal. Remember the inguinal canal that goes down when the testes descended into the scrotum. Okay? So just know the components of the spermatic cord, right? Which really simply is the blood vessels, veins and arteries, and the lymphatic vessels, the ductus deferens, okay, leading from the epididymis, right? And then surrounded by the external connective tissue, external spermatic fascia, then the cremaster muscle, the one that yanks up and down the testes, then another internal fascia, and then the blood vessels, the lymphatic and the ductus inside. That's the spermatic cord, okay, which is that, okay? And then you can kind of look at these structures later on, and so you can actually kind of visualize them. Okay, and then of course the urethra, which is split into this prostatic urethra, because the prostate is right underneath it's right underneath that bladder, all right? And then the membranous urethra that goes through the pelvic floor, and then the spongy urethra that goes through the corpus spongiosum, which is one of the rectal columns of the penis. All right? So just know the three divisions of the urethra, and then again, this one shows the spermatic cord. You see, you got the connective tissue, you got the cremaster muscle, then you got the internal fascia and all that, and then the blood vessels, lymphatic vessels, and whatnot in there forms the spermatic cord, and then of course corpus spongiosum down here with the urethra in it, and then of course the two corpora cavernosa. All right, so the penis is kind of another supportive organ with those three electoral columns, electoral tissue columns, all right? And it's got the head of the penis, the end of it is called the glans penis, all right? And the glans penis is covered by a cuff of skin, or an extended skin that covers the head of it, called the prepuce, okay? This is when, which is sometimes cut, okay? It's in a procedure done after birth, or when you reach puberty. It's mostly done for religious and cultural reasons. There's not real physiologic benefit to it. There are both a few benefits for circumcision, and some disadvantages. Okay, the guys who are circumcised tend to have more HIV infections, okay, than the guys who are not. Yeah, true. And then the guys who are not circumcised tend to have kind of local infections if they're not hygienic. If you clean it up, because when you close it up and you get all this stuff. So that happens. And then sometimes, testicular, penile cancer. Okay. But for the most part, it's definitely not like. So it's mostly done for cultural and religious reasons. There's no physiological benefit to it.
Now, for a long time, there was pretty much always done in the hospitals, wasn't it?
No.
Yeah.
Really?
It happens a lot.
I
I think it still happens
It's
a
purely
lot.
cultural. Really?
Now, when my kids were born, they asked us,
They are
do you want to cut or not?
What?
When my kids were born,
Yeah.
they said, do you want to cut or not? I said, yeah, chop that off.
Really? Yeah.
They
This
used to do it in the hospitals. I think they do it in a clinic afterwards, like now,
Yeah.
where you have to set up an
My
appointment.
kids were both done right after birth.
right after birth. Yeah. And when they do it after birth, they do it without anesthetic.
Yeah.
They just give them a sugar thing and they get it. But I just think it's
Never
it's
say-
personal reason. But
Oh,
anyway, it's I just think it's unnecessary. It's there for a reason. Everybody but it's there for a reason. But really, it's mostly done for religious and cultural reasons. That's all. Because either way is good. Okay, but
As long as you don't cut too much.
yeah, freaky things happen, and then you see this big penis, and then you keep asking, Mom, what were you trying to do, Dad? Come on, why didn't you just leave me well alone? I can make my own decision whether I want to be circumcised or not.
The Jewish people, they call it a bris.
Yeah, they do it. Some of them do it at puberty and so forth for other reasons, more cultural than anything. You know what I'm saying? But there are sometimes when it's medically necessary. When the foreskin is crimping too hard on the glans penis, when you retract your, the prepuce and it's really tight, it can cut circulation and so forth. It happens a few times. Then in those cases, it has to be removed so that the guy is free and not kind of strangled up like so. All right? So those are a few times when this foreskin kind of covers it up. So the two corpora cavernosa and then this corpus spongiosum, that kind of gives that clover-leaf structure also. And it's very evident when the penis is erect. When it's limp, you can't see these erectile columns because they're not erect. Okay? Corpus spongiosum carries the urethra. Okay? Erection. Okay? This is what happens during sexual excitement, and these electoral tissues become filled with blood. All right? And then the penis becomes rigid. it is necessary to insert into the vagina so you can deposit the sperm up, high up in the female reproductive tract. Okay? So that's what the penis is for. It's a copulatory organ. And then now we talk about the accessory organs that produces the glands. I mean, that produces the secretions that become semen. Right? So the seminal vesicles, they're posterior to the bladder. You know, they produce a fructose solution. Fructose is a sugar that is used for energy by the sperm cells. All right? And then, of course, when it joins the dilated vas deferens at the ampulla, and then it becomes the duct of the seminal vesicles becomes the ejaculatory duct. Okay? Where the sperm, the sperm cells coming through the vas deferens, right? Through the testicular secretions, which is about 4%, and then the seminal vesicles, which is the bulk of the secretions, about 65% of the secretions come from there. And then the prostate gland, which is right underneath the bladder. It's a donut-shaped, walnut-sized shape, about 4 centimeters wide, and so forth.
Walnut shape?
Yeah, it's kind of walnut-shaped, yeah, like a walnut, like it's sort of donut-shaped because it surrounds the urethra. Okay, but it's kind of walnut-shaped as well. Okay, kind of sort of like this. Okay, and then the urethra goes in the middle like that. So that's why the donut reference comes in. And then the bladder is right here. Okay, and then it goes, and then the penis is over here. Okay, and it goes to that side. And then, so this also produces, you know, some clotting factors that clot the clotting proteins from here to make the sperms thick and sticky. Okay, and you know, so it has glandular muscular tissue, and then the bulb of the urethra produces a mucus fluid which lubricates the penis, also makes the urethra neutral before the sperm cells come through. Right? Alright, so semen, of course, is a mixture of testicular secretions, seminal vesicle secretions, prostatic secretions, and bulbourethral secretions. Alright, so the testicular secretions make about 4%, and they carry the sperm cells from the testes. Alright, and the seminal vesicle, like I said, produces the bulk of the secretion, 65%. It's got fructose for energy. It's got these clotting proteins. Alright, and then the prostate then makes the proteolytic enzymes that break down these clotting proteins to kind of make the testes, the semen thick, and the semen is whitish, right? And the color comes from the prostate gland. The color of the whitish color of the semen is from the prostate. It gives that white, the secretions of it give the whitish appearance. Okay, and then the bulbourethral gland or secretes the mucus that makes the pH neutral, inside the urethra. Okay, and it also acts as a lubricant. It lubricates the head of the penis or the gland's penis. So physiologic function, we get the hypothalamus, which secretes gonadotropin-releasing hormone, right? Remember when we did the reproductive system, the endocrine system, and then it will then go to the anterior pituitary. The gonadotropin-releasing hormone causes it to produce the gonadotropins, which are FSH, follicle-stimulating hormone, and LH, luteinizing hormone. Okay, the luteinizing hormone is what we are calling also interstitial cell-stimulating hormone. Okay, ICSH. Okay, so ICSH, interstitial cell-stimulating hormone. So the gonadotropins are these two. The female also releases the same gonadotropins, but they have slightly different functions in the female. And then, like I was saying, inhibin produces by the testicular cells in a negative feedback fashion will inhibit FSH secretion so that you stop producing more sperm cells. Okay? Because you're always going to be producing testosterone because it's going to be maintaining the secondary sex characteristics for the male and doing other things. Okay, so when you've produced enough sperm, you can inhibit the FSH. You see, which is what goes to there. So hypothalamus, and then these are produced by the anterior pituitary. Okay? So we know about that. So what I want you to know is this hormonal control of the male reproductive function. Okay, so you know the hypothalamus produces gonadotropin-releasing hormone, and remember the hypothalamo-hypophyseal portal system connects to the anterior pituitary, which then causes it to produce LH and FSH, and LH and FSH are gonadotropins. LH causes the interstitial cells to produce testosterone. Okay, FSH causes the testicular cells, the ones that are nesting the spermatogonia and so forth, to produce more follicles, more sperm cells. Okay, right? That's number 3. And number 4, the testosterone will also cause the development of the sex organs and growth of the male and the muscular system and all that. And then number 5, the testosterone, once you've had enough in a negative feedback fashion, will inhibit the anterior pituitary and the the hypothalamus. Okay, these guys are waiting. So I think I will stop here, and then next time we will finish off the rest. Okay, when we talk about the reproductive system of the female. All right. Thank you, guys. Now we'll leave it here. So we just have to talk about the effects of testosterone and.
We're in a regular lab, right?
Yeah, we're in a regular lab. All right. Thank you, guys.